Hello everyone, my name is Rahul, and today I'll be going over a study by Eckert and colleagues titled Auditory Cortex Signs of Age-Related Hearing Loss. So these researchers wanted to learn more about presbycusis, which simply means age-related hearing loss. This progressive hearing loss affects almost everyone over the age of 70, and it disrupts normal communication due to impaired recognition of high and low frequency sounds in speech. Learning more about this hearing loss can help determine better interventions to help our aging population with this issue. So, this age-related hearing loss can be due to damage in the peripheral auditory system. For example, sustained noise exposure can damage the cochlea, which is exhibited by structural changes in cochlear nuclei and inferior colliculus. Spiral ganglion cells can also be damaged, causing this hearing loss. The researchers made a connection that this peripheral auditory system damage leads to changes in overall structure of the auditory cortex. To support this, previous studies show lower gray matter volume estimates in the auditory cortex of individuals with hearing loss. Ultimately, there were three questions that researchers wanted to test and expand on the knowledge from the previously cited study. First, does low and/or high frequency hearing loss show lower gray matter volume in the auditory cortex? Second, is there an indirect influence of age on overall hearing loss? Third, what are the gender differences in hearing loss? Now moving on to methods, the participants in the study included 49 older adults with a mean age of 69.58 years. There were 20 males and 29 females in the experimental group. The experimental group excluded patients with otological diseases and neurological diseases. There was a hearing test administered called pure tone audiometry, which is a hearing test used to identify hearing thresholds in patients. The participants then underwent one session of a structural MRI scan to examine gray matter regions of the auditory cortex. ROI analysis and VBM analysis are two analysis methods that the researchers used to make sense of the imaging data. In this study, the two different analysis methods complemented each other and showed similar findings. Now moving on to results, I'll also be referring back to the posed research questions. The behavioral measure in this case was the pure tone audiometry, which showed that the oldest adults exhibited the most high-frequency hearing loss. This refers to question 2, which described the age effect. The results also showed limited effects of low-frequency hearing loss, while there is a significant effect due to high-frequency hearing loss. And this refers to our question 1. The most significant result that the MRI scans showed is that after controlling for age and gender, there was a bilateral decrease in gray matter volume in the primary auditory cortex TE 1.0 region in patients with high-frequency hearing loss. This refers to our question 1. Another finding was that men had more high-frequency hearing loss compared to women. This refers back to our question 3. So in conclusion, the researchers found that the high-frequency hearing loss correlates with a reduction in gray matter volume in the auditory cortex of both aging men and women. Understanding that the brain region TE 1.0 of the primary auditory cortex is associated with high-frequency hearing loss allows for further research into treatments specifically targeting this region of the brain. Thank you, and please let me know if there are any questions.